we're ready to get it started. Well, good morning, everyone. Again, thank you for joining the last genetics grand round of the year. We do have a special guest speaker, which is Dr. Xi Lo. Also, he's the candidate for the DNA diagnostic lab faculty position. So we welcome him to join in-person visit and also do this grand round on the Zoom. Very quickly give you a little background for Xi's training, and he completed his bachelor from Nankai University in China in biology, then came to the US, did a master's degree in biochemistry in the University of Rochester and close to here. Then he went on and did a PhD in the biophysics in the Cornell University, also close to New York. Then he moved to the Baylor and did a postdoc under mentorship of Hugo Bellon, which is a very classical and excellent Drosophila geneticist. And also Hugo Bellon was the core director for the Undiagnosed Diseases Network Functional Genomics Center, which he made a significant contribution to multiple study, which I'm familiar with that nature of the study. Then he 2 years ago, a year and a half ago, and then he joined the ACMG, ABMG Laboratory Genomics and Genetics Traineeship at Baylor, which he is in the last, last 6 months of the training fellowship. Then he's interesting to the opportunity here for our DNA diagnostic lab. So with that short introduction, I'll let Xi kind of take the floor. Xi, go ahead.
Thank you so much for the introduction. And as first, I'm sorry that I didn't know why the camera on my laptop didn't work today. It worked well yesterday, so I'm connecting on my phone in the meantime to let everybody see my face and say hello. And then during my presentation, I will have my camera off. Okay, so let me start my talk. And thank you so much for inviting me for this presentation. And since this is a faculty candidate talk, I would like to introduce my previous work from 3 of my experience in UD and the Undiagnosed Diseases Network, in ClinGen, the clinical genome resource, and my diagnostic lab work from Baylor Genetics. So I would like to start my talk with Avery's story, which was reported on CNN News, titled "Mystery Condition Gives Way to Bright Future for 10-Year-Old." So since Avery's first visit with a neurologist at 5 months old, she was suffering from hypotonia, ataxia, global developmental delay, and progressive cerebellar atrophy for 10 years. Her family fell into despair when the doctor told them, "Science has not caught up with Avery yet." So the reason behind her condition was a complete mystery. In 2015, Avery joined the Undiagnosed Diseases Network. Our research and diagnosis in UDN shed light on a novel type of mutation, a toxic gain of function in a calcium channel gene, CACN1A. So her doctor then switched her medication to a drug that blocks the calcium channel. So I was so glad to hear her improvements under the new treatment. So from this story, I'm not only very proud of my achievements, but also realize how significant our work is to resolve patients' medical mystery and benefit society. So this is the central dogma of molecular biology that everyone is very familiar with. It explains the flow of genetic information within a cell between DNA, RNA, and protein. So using a similar design, I would like to propose here the central dogma of my career focus in medical genetics. So all my current and previous work is focusing on the critical questions between gene variants, genetic diseases, and the patients. So whether a gene is associated with a certain disease, whether a variant is causative, and how could we utilize those gene and variant knowledge for diagnosis to improve patient care. So how the researchers and clinicians all over the world are investigating those critical questions? There are 4 major steps in this process. Step 1 is the proposal of novel gene-disease relationships. Step 2 is the accumulation of genetic and experimental evidence over years. And step 3 is the validation of gene-disease relationship and variant pathogenicity. Step 4 is the application of knowledge and technology in laboratory for clinical case diagnosis. So I did 2.5 years postdoc work in Dr. Hugo Bellin lab at Baylor, working on Undiagnosed Diseases Network. So our UDN work has proposed many novel gene-disease relationships. The second step requires numerous years of efforts from clinicians and researchers over the world for the accumulation of diverse type of evidence. So for me, I received extensive training on diverse fields of research during my PhD and master experience. So after my postdoc, I worked with Dr. Champlon on a clinical genome resource, ClinGen, for about 3.5 years. And one major goal of ClinGen is the validation of gene-disease relationship and the variant pathogenicity. In the recent 1.5 years, I received extensive and comprehensive training in the molecular genetics and cytogenetics technologies in the diagnostic lab at Baylor Genetics as a LGG fellow, where I can apply all I have learned from previous experience to real clinical cases. So I would like to first introduce my postdoc work on UDN. The Undiagnosed Disease Network is a nationwide collaboration aimed to solve medical mysteries through deep science using advanced technologies. There are 12 clinical sites over the US recruiting patients. When a diagnosis is not reached, the UDN clinical sites submit the cases to the DNA sequencing core and the model organism screening center. So this is to show you the workflow of UDN. And I worked at the Drosophila core and the model organism screening centers. During my about 2.5 years UDN work, I provided functional evidence from model organism studies and structural biology analysis on 9 UDN cases, and 5 of them has been published, and I'm the 4th author on 3 of them. So I would like to first introduce one project, which was the Avery story. So from the whole exome sequencing, a de novo missense variant was identified. But this case was on hold for many years because it was very challenging for the physicians to explain why this patient displayed much more severe phenotype compared to previous known disease-associated variants in the CACNA1A gene. So you can see the progressive cerebellar atrophy from the neuroimaging, which is not detected in patients carrying other pathogenic variants of this gene. So the CACNA1A gene encodes the performing subunits of the calcium channel. There is one key feature of this structure that on every number 4 helix of each transmembrane domain, there are many positive charged residues which function as voltage sensor to control the open and close of the channel gate. So I discovered an interesting pattern of those positive charge residues and proposed the structural model to explain the special function of this variant as a Gale function. And then I performed the functional studies in Drosophila. I first generated 2 loss of function alleles in Drosophila. The wild-type genomic rescue construct can rescue the lethality, but not the UDN mutant. I then performed the electrophysiology in Drosophila eyes. This is the wild-type pattern. The on and off indicate the open and the close of the channel gate. So interestingly, the rescue with our UDN mutant displayed a significantly increased on and off, which suggests a Gale function effect. So consistent with what we see in the human brain, that the neurodegenerative phenotype only displayed in the UDN patients, but not the other patients. We also only saw the neurodegenerative result when the loss of function allele is rescued with the UDN mutants using electron microscopy. So both my structural analysis and the functional data in Drosophila indicate that this is a new type of toxic Gale function mutation. In another UDN case, collaborating with the Duke clinical site, we reported 3 families carrying dominant variants in a novel disease-causing gene, TBS2, with a syndromic cardiovascular and skeletal developmental disorder. So many genes in the T-bulk transcription factor family have been reported with human disease. So we identified the first association for the TBS2 gene. So we performed the Lucifer reporter assay in cultured cells and found the disease-associated variants diminish the transcription repressor function of TBS2. The mechanism is likely to be a loss of function through affecting protein production or causing protein instability. The overexpression of wild-type Drosophila or human TBS2 gene causes a strong developmental and functional defect in the Drosophila eye, and these effects are milder in TBS2 that carry patient variants. We also discovered a novel neurodevelopmental disorder, which is associated with rare de novo missense variants in RNF2 gene. So this new syndrome is named using our name, and my structural analysis was selected as the cover of the journal. So RNF2 functions as a ring domain E3 ubiquitin ligase, which is a core component of polycomb repressive complex. My structural analysis suggests the crucial function of this residue for the molecular interaction in the complex. The wild-type genomic rescue construct can rescue the lethality of loss of functional alleles in Drosophila, but not the UDN mutant. I also discovered a homoerotic transformation phenotype in Drosophila. In wild-type flies, the sex count only appears on the first pair of legs, but in this mutant, it shows on every legs. The wild-type RNF2 gene can rescue this phenotype, but not the UDN mutant. So next, I would like to introduce my previous work on the clinical genome resource ClinGen. ClinGen, which primarily funded by the NIH, launched in 2013. Currently, ClinGen working group and expert panels have a representation from 2,200 investigators across 56 countries. The major goal of ClinGen is to build an authoritative central resource that defines the clinical relevance of gene and variants for use in precision medicine and research. ClinGen is utilizing genomic and phenotypic data shared by patients, clinicians, laboratories, and researchers to provide expert curation or guidance for the improvement of patient care. So there are 5 main curation activities in ClinGen: gene disease validity curation, variant pathogenicity curation, actionability, dosage sensitivity, and somatic cancer curation. So I would like to first introduce ClinGen's variant curation effort. This is the evidence framework published in 2015 ACMG-F variant classification guidelines. It is the basic for the variant interpretation in most diagnostic laboratories. There are 28 coderial codes covering diverse type of evidence, and each code is defined at different strength level for a benign or pathogenic assertion. The final classifications are determined by the combinations of evidence code that apply. But what are happening in the field after 2015 guidelines, and how the sequence variant interpretation guidelines are evolving until today and to the future? ClinGen is playing a major role on this effort. So first, the 2015 guidelines are very general, but not gene and disease specific. Therefore, ClinGen recruits teams of experts in different clinical domains to develop gene and disease specific variant classification guidelines. Second, ClinGen has a sequence variant interpretation working group to provide general recommendations for the refinement and the evolution of ACMG-App variant classification guidelines. So now, in collaboration with ACMG-App and the CAP, we are developing the second version of the variant classification guidelines. ClinVar is the variant database that most diagnostic laboratories are using to report variant interpretations. There are more than 1 million ClinVar submissions from more than 1,000 submitters over 75 countries, mostly from clinical testing. ClinVar is utilizing a star system to indicate review levels. A 3-star curation is performed by an expert panel. So one specific goal of ClinGen is to develop a team of experts in different clinical domains to evaluate pathogenicity of individual genetic variants. So this is how our expert panel curations are displaying in ClinVar. It is shown as a 3-star curation reviewed by expert panel with a FDA recognized designation. You can also access the specification rules file on this page, and we also provide a link to view structured evidence in ClinGen's evidence repository. So we performed our curations in ClinGen variant curation interface, which is also available for public use. So I was one of the 3 ClinGen variant pathogenicity proxies and involved in the development and evolution of the curation interface. So currently, there are 55 ClinGen variant curation expert panels in various stages of approval. There are 22 expert panels have been approved and are submitting 3-star expert curations to ClinVar. Totally 4,431 variants have been curated across 78 genes and 53 conditions. So I was a member of a ClinGen expert panel application and approval committee, and I was leading many ClinGen expert panels work. Here, I would like to introduce one expert panel as an example to show you how ClinGen expert panels are developing gene disease-specific variant classification rules. The myeloid malignancy variant curation expert panel was launched in 2018 in collaboration between ClinGen and the American Society of Hematology. In 2021, this expert panel received the U24 grant support from NIH. So I was the leader, ClinGen liaison, and coordinator of this expert panel. And the scope of work of this expert panel is to curate variants in genes associated with the inherited risk for myeloid malignancies. There are 6 genes on the list, and we have published RUNX1 specific variant classification rules and examples of curations on blood ones and hematologica. We are now near the completion of GATA2 and the DDX41 specification guidelines. I would like to use RUNX1 as an exemplar gene and describe how we are developing specification rules. Gene disease specifications was based on the original ACMG app guidelines. I'm going to show you a few examples. BA1, BS1 at B9 quaternion for minor allele frequency is too high for a disorder. In the original ACMG guidelines, the cutoff for BA1 is 5%. According to the penetrance, prevalence, and the gene allele heterogeneity of the RUNX1 gene, we calculated the RUNX1 specific cutoff of BA1 and BS1. The gene-specific threshold has shown to facilitate the resolution of VUS and the conflicting interpretation variants to be none or likely none. PVS1 is the quaternion of predicted non-variants in a gene where loss of function is a known mechanism of the disease. And we developed a RUNX1 specific PVS1 decision tree for initiation codon variants and the nonsense or frameship variant at very N-terminal region. PVS1 is not applicable because different functional transcripts of RUNX1 gene use alternative star codon. For nonsense or frameship variant in other region, if it predicts to undergo nonsense mediated decay, it would have the PVS1 very strong quaternion. And we also define the exact region for the NMD prediction. The C-terminal region that are not predicted to undergo NMD would qualify for a strong level PVS1 quaternion because there is a very C-terminal motif that is known to be functional important. For some other genes, if the function of the C-terminal region is unknown, the strength of PVS1 may downgrade further. PS3 quaternion is for functional IC. Based on the data from published literature of RUNX1 validation control variants, we define the transactivation IC as a primary function IC with a cutoff of less than 20% of wild type. We also allow the usage of data from a list of secondary assay to assign different strength levels. PM1 is the code for mutational hotspots. We located all the reported germline RUNX1 mutations on the domain structure and defined 13 residues within the RUN domain as a mutational hotspot that affect DNA binding and or are somatic hotspots, which can qualify for the PMAM1 moderate level criteria. For other residues in the RUN domain, we will have a PM1 supporting level code. PP1 is a criteria code for segregation. We define the different strands of PP1 criteria based on the total meiosis observed within one or across multiple families. For example, there are 3 meioses in this category. So I would like to show you 2 variant curation examples. So this is a missense variant that has a VUS assertion in ClinVar, but our expert panel curated as likely pathogenic according to the following criteria. PM2 is met because the variant is a complete absence from population databases. The in silico prediction revel score is high for this variant, therefore PP3 is applied. PS4 supporting level is used because one per variant meets at least one of the RUNX1 phenotype criteria. One benefit of our ClinGen expert panel is that we can collect patients' data from multiple diagnostic laboratories, especially ClinVar submitters, to support our variant interpretation. So this variant is located in RUN domain and PM1 supporting is applied. PM5 supporting is met because a different missense change at the same amino acid has been classified likely pathogenic by our expert panel. So we described the application of our RUNX1 rules in 9 variant curation examples in the hematological paper, including this variant example. I also performed a structural biology analysis to support the likely pathogenic assertion. So you can see from the overall structure that the blue colored RUNX1 protein has 2 arms that clamp the DNA backbone, function like a tweezer. So the histidine 105 and the threonine 196 residues are on the tips of the 2 arms. They interact with each other through hydrogen bonding. So the mutation on H105 would disrupt the interaction and destabilize the complex. The threonine 196 residue is also a well-known mutational hotspot for pathogenic variants. So the next example is a missense variant that had a conflicting interpretation of pathogenicity in ClinVar. There were 2 pathogenic early submissions and 1 VUS, but our expert panel curated as a likely benign variant according to the following criteria. BS1 is met because the minor allele frequency is higher than our cutoff. BS3 is applied because experimental studies show normal function of this variant. The in silico prediction score is high, but based on our modified cutoff in version 2, this pathogenic criteria has been removed. I also performed a structural biology analysis. This residue, although located in the RUN domain, it actually is in a linker region and is far away from the DNA binding surface, which is consistent with our likely benign classification. I was also leading a ClinGen CDH1 variant curation expert panel. We recently published the updated CDH1 specific variant classification guidelines, which included 11 major modifications since the initial specification from 2018. Using the refined guidelines, 97% of variants with ClinVar conflicting interpretations were resolved to a non-VUS classifications. And 35% of VUS in ClinVar were also resolved. Overall, 88% of curated variants of CDH1 had non-VUS classifications. Next, I would like to briefly introduce ClinGen Gene Curation efforts. The ClinGen Gene Curation Working Group has developed a systematic framework to evaluate the clinical validity of gene disease pairs. There are 6 classifications used to quantitatively describe the strength of evidence for gene disease association. Genetic and experimental evidence is collected in different categories, and the points are assigned based on the strength of each evidence. And the final classification is calculated based on the point received. So the curation is performed in ClinGen Gene Curation interface, and I was also one of the 4 ClinGen gene curation proxies. I would like to show you an example of how ClinGen gene curation efforts are providing guidance for variant curation and the selection of genes on laboratories' clinical testing panels. There were 21 genes reported to cause Brugada syndrome, and they are included in gene panels from multiple labs. However, only 1 gene is curated as a definitive by ClinGen Brugada syndrome expert panel, and all others are refuted or disputed. Therefore, only the SCN5A gene is definitively associated with the Brugada syndrome, and the variant classification of other genes should be captured at the WES. ClinGen has currently curated 1,680 gene disease pairs across 48 gene curation expert panels covering diverse clinical domains. So I would like to briefly introduce a hereditary cancer gene curation expert panel. This expert panel is chaired by Dr. Champlon. There are 6 cancer genetics and oncologists in the group as expert reviewers. I was the leader, coordinator, and the primary curator in this expert panel and performed about 60% of curations. We also have many volunteer curators. The first effort of this expert panel was to curate the well-known cancer prediction genes for their major associated syndromes. We developed the list of genes starting with 114 cancer prediction genes identified in Dr. Rahman's Nature paper on 2013. 88 curations have been approved and published on ClinGen's website, and we are now near the completion of this effort. This is to show you an example of gene curation on the ClinGen's website. The BRCA2 gene is curated for both autosomal dominant breast ovarian cancer and the autosomal recessive Fanconi anemia. Both reach the definitive classification. So if you click the view report button, and it will show you the details of the curation. And we wrote a summary paragraph to summarize evidence used in this curation. The detailed points received in this curation and the PubMed IDs are listed in the matrix below. So in addition to gene curation and variant curation efforts, I was also playing important roles in many ClinGen central working groups. In the data platform working group for the improvement of software tools, in education working group for the development of curation SOP and provide training. I was also a co-member of expert panel review and the approval committee and the ClinGen oversight committee. So I would like to briefly introduce the sequence variant interpretation working group. The major goal of this working group is to provide to guide the improvement and enhancement of the ACMG variant interpretation guidelines. So we have provided general recommendations on many ACMG criteria. For example, I contributed to an effort to provide general recommendations for the application for the functional evidence PS3, BS3 criteria. The ClinGen SVI working group, collaborating with the ACMG app and the CAP, is working on the development of sequence variant interpretation guidelines version 2. You will hear more updates at the ACMG meeting next year. For example, a Bayesian quantitative approach is under development to calculate the pathogenicity of sequencing variants. So I plan to rejoin this effort after my fellowship. For the LGD fellowship in the recent year and a half, I received extensive and comprehensive training on molecular genetics and cytogenetic laboratory technologies. So here, I would like to briefly introduce the WGS test at Baylor and the project I'm working on. Comparison with the traditional WES and the CMA combo suggests the clinical utility of WGS as the first-tier genetic test. Whole genome sequencing, WGS, is the analysis of the entire genomic DNA sequence of a cell at a single time, providing a most comprehensive characterization of the genome. So WGS uses the next generation sequencing technology, and it enables large scale of sequencing of many short DNA molecules at the same time. This figure indicates the size limitations of different targeted or genome-wide diagnostic methods. It shows that molecular genetic technologies usually were used to detect smaller sets of genetic variations. In contrast, cytogenetic methods were used for larger rearrangements. In the recent about 10 years, many genome-wide approaches are widely used, especially whole exome sequencing and chromosomal microarray, but they also have size limitations and can only detect certain types of variants. WGS, in contrast, is a comprehensive test that can provide detection of almost all types of genetic variations. In this table, I'm showing you more details. The targeted tests on the left are only used for specific purposes. The CMA method was developed to detect copy number variations. Whole exome sequencing works best for the detection of nucleotide variants and small indels. CMA and the WES combo tests have been utilized as a genetic diagnostic testing over the last decade. However, the conventional phenotype-driven and stepwise approach can be time-consuming and often led to inconclusive results. In contrast, WGS offers the potential of a single test that captures nearly all genomic variation in an unbiased manner. At the BCM and the BG, we launched the WGS test in 2018, and we are the sequencing core of UDN. There have been already over 1,000 WGS cases tested for UDN, but I would not include those cases in the analysis for clinical utility. So we started clinical WGS test in 2019, and in August 2021, we launched the rapid WGS test with only 5-day TAT to facilitate usage of WGS as a first-tier genetic test. Our WGS tests have the following features. The average genome coverage is higher than 40x. And it is assembled to HG38. We performed SV, NV analysis, CNV analysis, AOH, UPD analysis from WGS data. And since last year, we started to analyze mitochondrial genome and the short tandem repeats. In the next couple slides, I'm going to show you a comparison between our WGS test results and the WES plus CMA concurrent tests, which were received within 1 month from each other in the recent years. I will compare them for case volumes, diagnosis rates, estimated cost and fee, required amount of DNA samples, turnaround time, and the variant types that can be identified. I'm comparing 4 clinical testings here. The green curve is for the regular WES and the CMA combo, and the blue is for the critical WES plus CMA, which has a shorter TAT. You can clearly see the significant decrease of case volume per month for the critical cases after 2020. The yellow curve is for the regular WGS cases. The case volumes keep increasing since it launched. For the rapid WGS, which had a 5-day PAT, it shows in red here, it started with very high case volumes and also increased this year compared to last year. For the diagnosis rate, you can also see here in the recent years, both the diagnosis rates of WES plus CMA combo test significantly decreased, especially for the critical cases. And in contrast, the diagnosis rate of WGS keep increasing, and the rapid WGS test shown in red here has almost reached 40% diagnosis rates this year. The significant increase of case volumes and the diagnosis rates of WGS in the recent year indicate that more and more people notice the advantage of a whole genome method and are considering WGS as the first-tier genetic test. Previously, the major limitation of WGS is its high cost. However, advances in the field of genomics over the past quarter century have led to the substantial reductions in the cost of genome sequencing. I got this data from NHGRI. Nowadays, labs can perform a WGS test under $1,000. Comparing to WES plus CMA combo test, WGS now has comparable estimated costs and fees. Another advantage of WGS is that it requires less amount of DNA samples. The WES plus CMA combo test need about 400 nanogram in total, whereas the WGS test only required about 100 nanogram DNA samples. This could be an important factor in certain situations when there is a limitation on the DNA sample collection. For example, prenatal tests or samples from specific tissues. In this slide, I'm comparing the turnaround time between WGS and the WES plus CMA combo test. For rapid WGS cases shown in blue here, the TAT mean is about 7 days, with more than 30% cases reported within 5 days. About 90% cases reported within 14 days. In contrast, for critical WES plus CMA combo in gray color, the TAT mean is about 14 days. None of the cases were reported in 5 days, and only about 44% cases were reported within 14 days. So note that this analysis only includes the WES plus CMA concurrent cases. If they were sent sequentially, their TAT would be much longer. So this data indicate that WGS can provide more rapid TAT than WES plus CMA combo test, which is especially important for urgent cases. In this table, I'm listing the variant types that were identified in the 61 solved WGS cases. There are about 75% cases shown in red was diagnosed with single SNV or CNV, which should also be able to detect it by WES or CMA. For the green colored, about 10% cases, they are the special variant types that can only be detected by WGS. For example, non-coding variants, short tandem repeats, and mitochondrial variants. So this is the well-known advantage of WGS test. But today, I would like to mainly focus on the categories in the middle of this table. First, the detection limit of CMA in the field is generally considered as a 10 to 100 kb. Therefore, I use 10 kb as a conservative cutoff to separate the yellow and the light green categories. In most labs and for most genomic locations, those 8.2% cases in light green color cannot be detected by WES plus CMA combo. For the cases labeled with probably, both SNV and the CNV were identified. And those cases require the combination of the WES and the CMA results for the diagnosis. For example, it could be an autosomal recessive disease with one allele CNV and the other allele SNV. It could be a dual diagnosis with both CNV and SA findings. Theoretically, those cases can be solved by WES plus CMA combo, but I will explain the caveats in case examples. In short, about 18% of the solved cases by WGS identify diverse variant types that very likely cannot be diagnosed by WES plus CMA combo. I would explain more details on those categories with multiple findings. For the WES and the CMA combo test, only one case was recorded as solved by the combination of WES and the CMA data for autosomal recessive disease. In contrast, there are 8.2% of cases in our WGS positive cases in this category. It could be because both WES and CMA would report their own findings separately as negative or indeterminate results. The physicians need to interpret the result themselves for the diagnosis. Therefore, we cannot pull them out as positive cases. Or there may be some diagnoses were missed. For dual diagnosis, the percentage is also lower than our WGS cohort. So this data indicates the advantage of WGS to diagnose cases with multiple findings of different types of variants. So let us look at the first situation. Problems when SNV and the CNV were detected from WES and CMA results separately on each allele for autosomal recessive diseases. Without knowing the result of the other tests, some variants may be missed or didn't receive enough attention as a high priority variants. Note that most labs do not report all the carriers in WES, especially when the phenotypes are not specifically related, and VUS is more likely to be missed. Second, if the cases need to be solved by the combination of both WES and the CMA data, it may require a good communication between the two diagnosis teams or may require data reanalysis, which is time consuming. It would be especially hard when WES and CMA tests were sent to different labs and were not sent concurrently. Third, WES and CMA are reported separately with negative or indeterminate results. A correct interpretation of the whole cases are missing. The first case example is a 9-month-old male with global developmental delay, failure to survive, short stature, hypotonia, hyperreflexia, GERD, dysphagia, and hyperspadias. I first worked on the CMA review of this case, and the CMA identified a 0.047 megabase heterozygous deletion on chromosome 10q. This region covered a gene named ALDH18A1. We located this deletion on the UCSC browser. It shows that this deletion includes most exons of this gene and can be considered as a complete loss of this gene on one allele. This gene encodes a bifunctional homooligomeric enzyme named P5ACS. There are 4 diseases associated with this gene listed in OMIM, 2 autosomal dominant and 2 autosomal recessive. So the first challenge for our variant interpretation is to decide which condition this variant is associated with. Should we report it as a causative for the AD condition, or it is just a carrier for the AR condition? I did a lot of literature study for the mechanisms. I will not explain it in detail here. In conclusion, although different syndrome representing a continuum of increasing severity of the same disease, P5CS deficiency, the dominant ALDH18A1 variants cause loss of function by dominant negative mechanisms. Haploinsufficiency is not the mechanism of the disease, and the heterozygous deletion alone is not sufficient to cause any diseases. We therefore, after a lot of literature reading and the study, finally reported the ALDH1 deletion as a carrier of the AR condition in the CMA results. So we made a conclusion from the CMA report. However, this case is still unsolved. Especially the concurrent global maps test results show the reduction of the urea cycle immediatase and also altered several TCA cycle analyzed. So although it is not conclusive, but it is consistent with the P5CS deficiency and should not be seen in a carrier status. So therefore, I also took over the review of the WES result of this case. So I did find a missense variant which was shown as a homozygous in the WES results. The father is the carrier and the mother is negative. So it is very unlikely that the proband has a de novo mutation at the exactly same location. It is hard to explain this finding only based on the WES data itself. But since we know there is a deletion of this gene from the CMA results, this variant actually is a hemizygous variant on the other allele, and the mother should be the carrier of this deletion. So no previous reported evidence for this variant, and we report this missense as a VUS for indeterminate finding in the WES report. So I changed it from homozygous to hemizygous variant in this report. Interestingly, while I'm preparing for this talk, I noticed a very recent paper just published this September, which supports the pathogenicity of this missense variants and further confirmed our diagnosis of these patients. In short, they identified homozygous of this missense variant in 4 affected probands from 2 unrelated families that exhibited variable phenotypes of the P5CS deficiency. They also performed functional studies which showed the impaired P5CS function. With those new evidence, we could probably recurate this variant as likely pathogenic, and it further confirmed our diagnosis. I also analyzed this residue on the crystal structure during my curation. This stranding contributes to a hydrogen bonding interaction with a nearby asparagine, and they are also very close to the DNA binding pocket. It also indicates the possible functional importance of this residue. So now let us go back to our major topic of today. And if this case had been tested by WGS, the results of those 2 variants would have come out at the same time on a single test. And first, it will save us a lot of time and effort for the interpretation of the variants. Without knowing it has a biomolecular variance, we had to find out the detailed mechanism of the gene and decide AD or AR condition to report. Second, it would now require a high interaction between the data of different tests. So without knowing the deletion on the other allele, WES itself would mistakenly interpret this missense variant as homozygous. The variant, the VUS, may not be pulled as the top candidate variant. And third, a comprehensive interpretation will be reported. Currently, both WES and CMA reported their results as indeterminate finding. So, okay, next, let's look at the second situation. And problems when SNV and CNV were detected from WES and CMA separately as a dual diagnosis. So first, if the test were sent sequentially, people may think the case is solved and will not send the other test. Therefore, the other diagnosis will be missed. Be careful that the case may not be a phenotypic expansion or expression variability as you thought. There could be a second diagnosis you miss. Second, WES and the CMA are reported separately with their own positive results. A comprehensive interpretation of the whole cases are missing. Third, complex diseases that involve in multiple genes could also be missed. So the second case example is a 15-year-old female with abnormal cerebral white matter morphology and leukodystrophy, sensory neural hearing impairment, malnutrition, failure to survive, growth delay, and behavior abnormality, GERD, global developmental delay, abnormal foot morphology, delayed speech and language development. In 2020, CMA identified a copy number loss of the most distal part of the chromosome 22q, spanning about 0.875 megabase in size. Loss of this region is associated with the Phelan-McDermid syndrome. This syndrome is characterized by the neonatal hypertonia, absent to severely delayed speech, development delay, minor dysmorphic facial feature. The Shanks region is the cause of this disease, and the ring chromosome 22 was reported. On the gene reviews, it is also noted that normal stature and normal head size distinguish this syndrome from other disorders. In our patients, the diagnosis of Phelan-McDermid syndrome cannot fully explain all the phenotypes of these patients. We noted worsening tongue, central nervous system findings, especially leukodystrophy and failure to survive, that are not consistent with this syndrome. So I'm showing you here the timeline of the diagnosis test performed for this patient. Back in 2015, chromosome analysis identified the ring chromosome 22, which is consistent with the 22q terminal loss detected in CMA 2020. Because of the additional neurological findings, they also sent multiple biochemical tests for these patients, as well as an in vitro leukodystrophy panel. It was still undiagnosed until this year. WGS identified an additional disease, mitochondrial DNA depletion syndrome 1 MinGTAP. It was also confirmed by concurrent global maps and plasma thymidine determination tests. So this is the result of the WGS test. We also detected same deletion that was found in CMA. In addition, a missense variance on the other allele within the same region was also identified. Similar as the case 1 example, this is also a hemisex variant in autosomal recessive disorder. Totally 37 protein coding genes are in this deletion region. SHANK3 is the only haploinsufficient gene. There are also another 6 autosomal recessive genes in this region that would not be reported in CMA results. TYMP is the gene we found the hemizygous missense variant on the other allele. The major clinical features of MING-GE are GI issues and the local encephalopathies, which was the major concern in our patients. This variant was previously reported in homozygous stage in a 29-year-old Turkish male who was diagnosed with MING-GE. It is absent from NOMAD, the Revel prediction score is high, and we curated as the likely pathogenic variant. I also perform a structural biology analysis. This leucine residue is located at the hydrophobic core between multiple alpha helix. The purlin mutation would predict to disrupt the alpha helix structure and the hydrophobic interaction in this region. And many other leucine to purlin variants in this region were also reported in MING-GE patients. The TYMP gene encodes a thymidine phosphorylase. The deficiency of this enzyme will cause the elevation of thymidine and the dioxyuridine, and therefore result in mitochondrial dysfunction. The global maps and the plasma thymidine determination results show a significant elevation of thymidine, which further confirm this diagnosis. This is the global maps data compared to previous cases that detected thymidine and the dioxyuridine. This case in red dots showed much higher abundance of those two metabolites. So in summary, this case is a dual diagnosis and also include an autosomal recessive disease with both CMV and SMA findings. It was not fully solved for many years until WGS was performed. Another very quick example is that there is a dual diagnosis for autosomal recessive disease with Miller syndrome. The mitochondrial diagnosis will be missed with a WES plus CMA combo test. Okay, at the end of my talk, I would like to go back to the central dogma and propose my future career plans. First, utilize all my previous knowledge and expertise. My major focus would be to contribute to diagnosis of clinical cases as a lab director, collaborating with the clinical team. Second, while I'm working on patient cases, there could be a feedback loop in this central dogma involving in some medical research opportunities. For example, if there are any interesting gene variants from patients, I could utilize the strategy and resource from UDN for the discovery of novel disease-causing gene variants. I also want to involve more in ClinGen activities for the development and evolution of curation guidelines. And there could be also other biomedical research possibilities. So I would like to thank all the members in Dr. Hugo Ballon lab at the Drosophila Core of Model Organ Screening Center in UDN. I would like to thank all the members in myeloid malignancy variant curation expert panel. I would like to thank all the ClinGen PIs, working group chairs, and the key coordinators. And I would like to thank the clinical diagnosis team members at Baylor Genetics and other current LGG fellows. Okay, yeah, I'm done my talk and go back to my camera on my phone. And
that's a great talk, Xi. Thank you for sharing the extensive experience of training you have. That's open for question. Feel free to unmute yourself, raise your hand, or you can type it into. And just remind you, the CME code is in the chat. It's 34325. If you need it, please do so. question to anyone? I could start it. So I think your training on the biophysics and the protein structure model is definitely very, very unique. I guess from your own experience, how consistent sort of your based on the DNA and then come to the structure prediction, and then you have a function study you are good at to interpret the function study. Do you, like in your experience, how consistent that from your own experience versus like a systematic to evaluate large scale of the variant for this content?
Yeah, that's a great question. First, I'm quite surprised that my previous experience of structural biology is very useful background throughout my each experience. You can see different cases and different variant interpretation can be used. So, and I think functional evidence and also like the structural model is an important future field in this area for variant interpretation, but currently it's not widely used because when we interpret pathogenic, for benign side, the most useful tool is a population database, and for allele frequency for pathogenic side, most useful evidence is the patient's case data, which is limited, how many patients reported and collaborating between labs. And the second one is a functional study. So currently it's only limited to specific researchers for some different purposes. For example, only single variant means for different research literature, but not very few high throughput functional assay, including the structural analysis, was performed in the field, and it's limited the variant interpretation. Therefore, a lot of variants stay at the WES. So for me, my special expertise on structural analysis is mainly based on the known structure, on crystal structure, and it's a varied, the impact varied from each situation, each residue. This is my special expertise. I think it cannot be widely used in the field of diagnosis and into the guidelines. And mostly some specific cases of research, it would be helpful. And but some other like structural modeling, computational prediction, although it's not such reliable as the individual analysis, but in the future can be possibly applied in the high throughput analysis as some kind of evidence. Yeah,
that's great. The follow up on that one, how much from the ClinGen curation, like the structure prediction, like machine learning or AI has been incorporated this process so far?
Yes, so I think this maybe about 1 month ago, ClinGen published a new paper to evaluate the different in silico prediction tools. And because in the original 2015 guideline, the prediction tool is a supporting level PP3, and they proposed in some certain situation could be have a higher like a moderate or even strong criteria for the prediction tools. So my personally, I think it's currently still limited the prediction, and because my personal experience for pathogenic variant, they usually can have a very high prediction score, but the false positive rate is very high. There's a lot of benign variant also get a very high prediction, and different tools have like a different rationale and the machine learning method to make the prediction. This area is still developing. In the future, I hope there could be a better and better predictors. And currently, we already have better predictors like a Revel, and they use is a mega predictors combine 13 different other predictors results. So already be better than previously people use polyphony sieve. So now it's already better. And also for splicing prediction, now we have splice AI perform much better than previous predictor. It's getting better, but still it's only like a small piece of evidence for variant interpretation field. To me, I think if it's not like a severe variant, like a nonsense frame shift, only based on the prediction, especially for missense variant, we cannot make it a pathogenic or likely pathogenic conclusion only based on prediction. Still want to see the real evidence from functional assay or the clinical case data. Yeah.
Well, thank you. Mohammed, your hands up. Go ahead.
Yes, I have a question. Would you mind highlight how did your lab achieve turnaround time 5 days for rapid whole genome sequencing?
So how it's reached? It just because for whole genome sequencing, yes, there's more some technical details. And for example, it's there's one of the capture step, and for the compared to the WES, and also our lab try to like optimize and make every step the fastest as possible. I'm not sure about what's the PAT here, but I think in the field it's a yeah, 5 days PAT is, I think it's proposed the maximum PAT, but depends on each case. For example, if we need like because any confirmation test still may take a longer time, especially for like sometimes there's a parental samples is like needed. Sometimes need to run parental samples as a lower coverage, and so to facilitate the process. So I'm trying to make this process faster to make the especially for NICU and urgent cases. Yes, still there could be case specific. Yeah.
So Mohammed, just for your information, so our DNA lab also offer rapid whole genome for 5 days here at Yale too. So
okay, yeah.
So yeah, it never takes 5 days. I mean, our average turnaround time is 3 or 4 days.
Great.
That's
wow,
great.
that's impressive.
Yonghui, can I have a question?
Yeah. Go ahead. I think we have get the last question.
Yeah, excellent talk and really cover a lot of. I have one question about the 22q13 deletion. After you do the whole genome sequencing, do you give any detailed deletion-wise, like is that deletion exclude the telomere, or they still have telomere segment there? Or, because usually we think this 22q13 deletion is mostly terminal deletion, but by genomic sequencing probably can reveal actually the deletion is still maintain the telomere and the subtelomeric region too. That's part of that. If you have a whole genome sequencing, you probably can give more detailed structure for the chromosome scale. Do you have any information about that?
Yeah, maybe need to. That's a great question. I may need to go back to check details from the result here, because I didn't make that into slides here. I have slides from the CMA result include the detail of the region, and I think that this region is to have a deletion. I assume it's include the terminal. The deletion includes terminal region, but.
We usually call it terminal deletion. As you mentioned, actually, this deletion involved like over 30 genes, and another gene more toward the proximal part is the PAC2 genes related to Parkinson's disease, and I think this is also pretty challenging for interpretive result. Once we get into the PAC2 gene, you will consider that could like relate to the neural deficiency. The thing is like your story mentioned like maybe there's a variant in the gene and then reveal some recessive condition. So I think that's advantage. You actually have a whole genome sequence, and then you can reveal the deletion at the same time, reveal any like variance in the gene, and then see any contribution to the phenotype.
Okay, thank you.
All right, so we are about time, and thank you everyone joined. Thank you, Sis, for a wonderful talk. And then hope everyone enjoy the holiday and Happy New Year. We return after New Year.
Thank you.